we're basically on time, so yes. Who will tell us about a sparse mapping from structure to function in microbial communities? And I'm looking forward to the talk. The floor is yours.
Thank you, guys. What a great conference and great first few talks here in the session. Really thought Jeff's introduction was superb, and it really, I sort of share his view about how to approach these problems. And I'd like to tell you sort of some new work that we've been doing in my lab over the last couple of years along those lines. So just to back up to the beginning, if you look at life on the planet, one of the sort of striking features of life on the biosphere is that they drive and participate in these persistent nutrient cycles that are taking place on a global scale. And Jeff sort of mentioned that microbial communities play an important role, and I think it's I would almost even go further and say it's almost hard to overstate the role that microbial communities play in these nutrient cycles. And what's happening is that these nutrient cycles are driven by redox reactions. And bacteria and other microbes are harvesting energy from those redox reactions and occupying metabolic niches which are defined by those reactions. And so I think it's really important to think about these communities in the context of this global process. And it's also important to understand that this is a sort of a co-evolving system, that the microbial communities are themselves influencing these nutrient cycles, sustaining these nutrient cycles, and then being influenced by those cycles as well. And so just to really sort of drive home this point about the importance of these communities for sustaining this global flux of nutrients, if you look at this sort of left half of the nitrogen cycle here, which is the transformation of nitrate to dinitrogen gas, this happens almost exclusively in microbial communities and bacterial communities in soil. This process is called denitrification. And so these communities are the evolved systems that are driving this flux of nutrients. And in a similar example, which was alluded to already, the fixation of inorganic carbon, carbon dioxide, to organic carbon, roughly half of the carbon that's fixed on the planet is done by microbes. That's about 100 gigatons per year, which dwarfs by a factor of 10 the amount of carbon that's produced by all human activity, which is about 10 gigatons per year. So I think when one thinks about these microbial communities, it's important to put them into this not only ecological context, where they are performing these metabolic transformations in order to sustain themselves, but also to understand these as evolved systems that have over billions of years come to be able to perform and sustain these chemical transformations. And so I think understanding how these communities are structured in order to drive these transformations and how evolution has built these systems over the history of the Earth is really a compelling question. And so that's the question that my lab is really focused on these days. And just to sort of more sharply articulate that question, what I'd really like to be able to understand is how the mapping from the genomic architecture of these communities to their metabolic function works. What is the code which can tell me how the structure of a community at a genomic level is mapped to this emergent flux of metabolites through the community? So I think this is an important question for the reasons that I just argued. And I think we, of course, this is not a new question. People have been working on this problem for a number of centuries, probably, maybe the last 150 years, since we really appreciated that the microbes are playing such an important role in this process. So in the last decade or so, what the sequencing revolution has sort of made it clear that there's probably some relatively simple relationship between the genes that a community possess and their metabolic function. And this is exciting because it lends itself to the idea that there may in fact, despite the complexity of these communities, there may in fact be simple rules for understanding how their metabolic function emerges from the sort of immense complexity that you guys have been discussing in the last hour or so. So just to give you a couple of examples of these hints, there's a recent study, the Tara Oceans Project, later analyzed by Luca and Dobely, and what they found was that the functional gene content of microbial communities in the ocean is more closely related to the metabolites that are present in those communities than is the taxonomic structure. So the implication or the assumption here is that that functional gene content is influencing the environment of these communities and the converse as well. Sort of a similar result has been floating around in the human microbiome world for about a decade now, which is shown here on the right. And the idea here is that the functional gene content, that is the pathways that these communities possess, may be sort of conserved quantities in these systems, but the taxonomy, again, can be highly variable. And so I think in light of this question of mapping genomic architecture to metabolic function, these studies are suggestive that there might actually be some sort of simple relationship between these two quantities. But the problem with these studies is that it's a bit hard to extract that relationship, in part because it's hard to tell how much of these results are driven by the local environment in these very complex natural environments, and how much are actually due to the functional relationship between gene content and metabolism in these systems. And so what we would like to do is to sort of take this observation and this big picture question into the laboratory and ask a sort of sharply defined quantitative question. We know there are these associations between gene content and properties of the environment in the wild, but what we would like to know is quantitatively, how much through a community, how much of that information is encoded in the genomes of the organisms in that community? You can imagine that the answer would be not much, right? Although the hints that I just showed you suggest that might not be the case. And the reason is that, of course, these genomes encode these phenotypes of these organisms, and those phenotypes then give rise to interactions, which we just saw a very nice talk on. And those interactions then ultimately give. So we would like to ask this quantitative question, and the way we're going to pose it is as a prediction problem. If I give you the genomic structure of a community, can you predict for me its metabolic function? That's where we started with this project. And the approach we're going to take is what I would call a statistical empirical approach. And we're going to use this process of denitrification, which is so crucial to the global nitrogen cycle, as a model system. And the approach is important. So the first thing that we're going to do is go into the wild and isolate a diverse library of strains. And the reason that's important, of course, is that when we look at the structure of these communities in the wild, there's a massive amount of genomic and taxonomic diversity in these systems. So we need to try to capture that in some sense in the laboratory. But then coming into the laboratory, the goal is going to be to quantify the metabolite dynamics, which is something that's obviously much more challenging in the wild. And that's going to set up a quantitative attempt to map the sequence variation of those isolates to this metabolic function. So I think these ingredients are not chosen by accident. It's really important that we capture this genomic diversity in order to make what's going to ultimately be a statistical mapping from sequence variation of this system to this metabolic function, which is, of course, the evolved property of this system. Okay, so I want to take a brief step back and give you some introduction to the system before I sort of dive into the results. So I've come to realize over the last couple of years that denitrification is really a fantastic system for asking this question. And it is now clear to me that choosing the right system has many payoffs. So denitrification is the process of transforming nitrate to dinitrogen gas, as I just mentioned. And this happens in the gut, it happens in your teeth, it happens in soils. And what's happening is these bacteria are using these nitrogen compounds in place of oxygen as electron acceptors. So they're not incorporating the nitrogen into biomass, they're donating electrons from reduced carbon, and that is driving their production of energy. So in the wild, this is a collective process. So you'll notice that this is a cascade of reactions from nitrate to nitrite, nitric oxide, nitrous oxide. And what happens in the wild is in some cases you can have one strain that just does all four reactions. And in some cases you can find communities that are built in such a way that you have one strain doing the first reaction and another strain doing the subsequent reaction. The other advantage of this system is that the molecular and genetic details are known almost to the level of, say, chemotaxis. And the other interesting and important thing is that this is taxonomically broadly distributed. So these genes have been subjected to HTT for a very long time, and they're distributed broadly across the bacterial kingdom. So it's not that there's just a few strains that do sort of photosynthesis, for example. Okay. So in following this roadmap that I outlined for you here, what we're going to do is we're going to go into the field and isolate a library of isolates that perform this metabolic process. And we're just going to focus on the first two steps of this cascade. And the reason is that these metabolites are themselves soluble and quantifiable, whereas these are gases and a bit more messy. And the library of isolates that we constructed consists of three phenotypes. We've got types of strains, that do the first two steps. We'll call those NAR-NER strains after the enzymes that perform these reactions. We've got strains that perform only the first step. Those are called NAR strains, and strains that only perform the second step. Those are called NER strains. And of course, red plus blue makes purple. So the color scheme makes sense. So my postdoc, Karna Gowda, went into the cornfields of outside Urbana, Illinois. I recently moved to the University of Chicago, so we imported strains from downstate. And he isolated them and phenotyped them in the laboratory. So here's the phylogenetic tree of the 60 isolates that he brought into the lab. And so you can see it's a few important things to note, that these phenotypes are broadly distributed on this phylogenetic tree of proteobacteria. So it's not that all the ones that perform the first reaction only are located in one section of the tree. And so what Karna can then do is he can go into the lab and quantitatively measure the metabolite dynamics, similarly to what you saw in the last talk, but in this case for 60 isolates in parallel. And let me just walk you through one of these plots because it's not entirely obvious what we're looking at. We're not looking at abundances, we're looking at metabolites. So the vertical axis is the concentration of nitrate and the concentration of nitrite in blue and red. And so if I have a strain that performs both of these reactions, what I see is that it eats up the nitrate, it produces some nitrite, and then consumes that nitrite, and then the situation is over. This is just a batch growth. Now, if I have a strain that just consumes nitrate, but not nitrite, nitrate is NO3, nitrite is NO2, of course, I consume the nitrate and produce the nitrite. And if I have a strain that only does the second step, I just get this curve, okay? So what we can do then is parameterize one of these very simple models. This is a model very similar to the models that Guy and Ned, Pankaj, and all of our friends are studying theoretically. And so this is a very nice model, I think, because it will give us quantitative predictions, which we'll be able to test going forward. So what we do is we take these data points, the blue and red points here, we infer a consumer resource model, which describes the abundance dynamics X, the nutrient dynamics A for nitrate and I for nitrite. And we just have simple Monod consumption and production terms. And so there are just a few parameters: the rate at which these strains convert one species to another, the biomass yield, that's how many cells per unit nitrate they consume that they produce, and then an affinity parameter, which we can't properly constrain from our data, so it's just fixed, okay? So essentially, these dynamics for each strain are now encoded in a set of scalars. That is the rate of nitrate transformation and the biomass yield per nitrate molecule consumed. And these models do a very good job of quantifying the phenotypic metabolic variation in this library. So after a big effort between Karna and a former undergrad in the lab, Derek Ping, what we've done is we've quantified the rates and yields of nitrate and nitrite transformation for all 60 strains in this diverse library that I showed you a second ago. And so you can see each one of these sort of rows comes from a set of graphs like this. And I can just read off here, this is the rate at which the first strain performs nitrate reduction, and so on. And so now I've taken this diverse ensemble of strains from the field into the laboratory, and I've written down their phenotypes in terms of just a few model parameters. Okay, so I've got this sort of phenotypic quantification part done, but ultimately what I would like to do is to relate that phenotypic variation back to the genomic variation in these communities. And to do that, what we did was, of course, we whole genome sequenced each one of these strains. And what I'm showing you here is simply the gene presence absence in the denitrification pathway for each one of these isolates. So this matrix is just a binary matrix. And the colors are telling you something about the function of each one of those enzymes. So the blue ones perform the first step in the reaction, the orange one's the second step. These are regulators and transporters. Okay, so because the molecular details of this pathway are well understood, we can annotate these genomes and look at what each strain has. So here's where we took what I would call a leap of faith, which is to conjecture essentially that the knowledge of the presence and absence of genes that each strain possesses is going to harbor substantial predictive power about the metabolic phenotypes that each strain displays in the lab. And there's, one could say, well, this isn't going to work because, the promoter architecture is going to be different, even though these two strains maybe have the same gene. Or there's going to be some other regulatory component that's not going to be accounted for here. However, I think, there were hints in the literature that gene content has a lot of information about this community metabolic function. And so our thought was, gene content is perhaps strongly predictive of these metabolic phenotypes. And if that's so, perhaps that mapping is simple. And so the proposal that we made was that if I take this binary vector of gene presence and absence as a proxy for the genotype of each isolate in the library, so this is a binary vector of zeros and ones, what I'm going to do is I'm just going to, by linear regression, try to predict the phenotypic parameters for each strain from that binary vector. So this is sort of the simplest possible one-point model. And the idea is that that for some strain I that has gene J, beta J will tell you the impact of having that gene on that phenotypic parameter. Okay, so if beta J is high, you get a big boost in the rate at which you do nitrate reduction if you have that gene. And so these are observed quantities that are measured in the laboratory. These we've sequenced, of course. And this is not a straightforward regression problem in the sense that we have, too few data points and too many independent variables. And so what we have to do is regularization. Okay. So when we do that regression, remarkably, it works quite well. And so what you find is that here I'm showing you the observed biomass yield on nitrate and the predicted biomass yield on nitrate. The R squared is about 0.75. The out of sample R squared, meaning if you gave me a new genome, is about 0.5. And the remarkable thing here is that this prediction is relatively sparse in the sense that all I need is knowledge of a few genes in the pathway to get a lot of predictive power about the metabolic phenotypes of these strains. And so this works, these independent regressions work for all 4 of the phenotypic parameters that we quantified in the laboratory. And I think the important thing to realize is that if we didn't have this diverse library of isolates from the wild, we wouldn't have sufficient phenotypic variation on our x-axis for this regression to be able to explain, right? So I think that's an important lesson to take away from this work. So as I said, this regression works on all 4 of the phenotypic parameters, and here's another example. So one might ask themselves, why does this gene presence absence have so much predictive power? It doesn't make any sense. Maybe you guys are fooling yourselves. I think what we're starting to realize is that these regression coefficients actually are uncovering conserved mechanistic features of the pathway. And so here's an example. There are actually two enzymes, NERK and NERS, that perform this second reaction. And in vitro and in vivo, it's known that NERK is much, much faster than NERS, like a factor of 100. And so what our regression shows is that if you have NERK, you perform this reaction more quickly. So that's consistent with physiological data on model organisms. And so I think what we're seeing is that there's a conserved, an evolutionarily conserved, I would argue, property of these enzymes, which now being reflected in the phenotype and the phenotypes of these libraries. So we've gone from genomes to phenotype with this simple gene presence absence picture. And but I haven't said anything about the community, right? So at this point, we're a bit unsatisfied in the sense that we haven't made any predictions about community level metabolism. So, but the model gives us a really nice way to make predictions and then test those predictions. So here I'm showing you the metabolite dynamics for one of these strains. This is a NAR-NER strain, and another strain, just a NAR strain that only does the first step in the cascade. And so what I can do is I can take the models parameterized on monocultures and do just what Caroline was doing in the last talk, make a zero free parameter prediction of the metabolite dynamics in co-culture. I just integrate these models together, I get a curve, and then I go in the lab and I make a measurement, and by and large, it works remarkably well. So these are the metabolite dynamics in co-culture predicted solely from information on monoculture, and that prediction, I think, works surprisingly well. So we've done this type of experiment for all possible combinations of 12 strains in the library that have each of these three phenotypes. And the blue regions of this heat map are just telling you that this prediction works, okay? Now the yellow region of this heat map is telling you that this prediction is not working. And the really striking thing that we observe here, which is quite fascinating actually, is that this prediction fails when you have a NER strain, which does the second step, in co-culture with a NAR strain that does the first step. So when you culture strains that have the pathway split up into two genomes, you see that this prediction is just a disaster, right? So these lines are our prediction and the points are the data. It's not working. And so this is something that we're looking into. We think it might actually have something to do with pH. That pH is that low pH is the niche that's occupied by these strains, and that's a story that I can discuss with you guys after. So, but by and large, this is the situation we're in. The prediction seems to work quite well, and one could then ask, can I go higher? Can I go to 3 species communities? And the answer is, if I have 3 species communities that do not have one of these NAR plus NER pairs where the prediction fails in the pair culture, by and large, my predictions work quite well. If I have a 3 species community that contains one of these pairs, then my prediction fails again, as you might expect. But I can take a coarse graining strategy where I treat this pair as a single effective species and use the pair culture information, and now my prediction starts to work again. So there's a sense in which this NAR plus NER pair is acting as an effective single species in the community. And this is just showing you that if we take this sort of coarse graining approach to predicting the metabolite dynamics in the 3 species community, that our error in the prediction is very low. And if we don't, for some of these communities that contain these pairs, the prediction fails. I am going over time, I'm sorry. I just want to make one sort of high-level statement here at the end, and that is the conjecture that in the search for these simple relationships, these simple ideal gas laws for microbial communities, should they exist, I would like to conjecture at least that that genes and metabolites are potentially the good variables for finding those simple relationships. And I think what we've shown here is that a very coarse characterization of the genomic architecture of these systems allows you to predict these phenomenological phenotypic parameters and then the emergent metabolism of these systems. And so I would like to continue this sort of line of thought, and I'd love to talk to all of you about your thoughts in terms of finding these good variables, because I think this is really a key problem. One last point, which is, you might say to me very justifiably, hey, you've got 3 species in your community. okay, maybe there's just not enough complexity. And I have a separate study, which I unfortunately don't have any time to tell you about today, where we're studying these self-sustaining closed microbial biospheres that are immensely complex. And in these systems, we're starting to see similar things where the emergent metabolic structure, metabolic function of the community, which in this case is carbon cycling, is relatively simple despite the taxonomic and population dynamics in these communities being quite messy. And so my thought is that, again, the invariance in these systems is the gene content and the metabolite fluxes. And so I'd like to try to push that idea forward in the next couple of years. So just to close up, I think we've shown that the metabolite dynamics are sparsely encoded in the gene content, and this opens up a host of exciting questions. Can we design communities? Can we understand the evolutionary principles behind this? And can we look at the structure of communities in the wild? This work was done in large part, almost entirely by an incredible postdoc in my group, Karna Gowda, and he is co-advised by a good friend, Madhav Mani, and I, and both of those guys have been just such a pleasure to work with. So, oh, and the last thing is I'm recruiting postdocs here to UC, so please get in touch.
Excellent. Thank you, Seppi, for a fantastic talk. We have a bunch of questions in the chat, so I'll start. Shoal Pollack is asking, did you check if, for instance, there is correlation between combinations of genes and promoter architecture? And then separately also, perhaps for structure in which betas are non-zero under different conditions for the monocultures?